Good morning everyone. In today's video, I'll be talking about growth. Now, what is meaning of this growth? Growth means usually there is an enlargement or increase in the cell or the size like that. But in case of microbiology, the growth means increase in the number of cells in a population. Okay, this may be in case of bacteria, okay, or actinobacteria or cyanobacteria, whatever it is. So basically in case of bacteria, growth means there is an increase in the number of cells in a population is known as growth. Now there are various steps in the growth. The first one is total 4 steps, that is lag phase, log phase, or also known as exponential phase, stationary phase, and death, where the cell stop their growth. These 4 are very important steps. Okay, there is no other steps or any kind of a modification right now. These 4 are conventional steps in the growth of any kind of a bacteria which is growing in batch culture. Now, what is batch culture? Batch culture means, in case of microbiology, in case of bacteria, that is microbes or bacteria, I should say bacteria, they are growing in a closed culture vessel with a single batch media. Okay, and if we want to calculate or measure the growth of any kind of a bacteria in batch culture, we need broth, or it is also known as liquid media, not agar media, because in agar media we cannot measure the growth of any kind of a bacteria which is present in the plate, that is in a agar media. But in case of broth, which is a liquid media like nutrient broth, if we are growing E. coli in nutrient broth, we can easily measure their growth, how fast they are growing or how slow they are growing, like that. So we need the broth or liquid media for their growth. Now this is a batch culture. Another important thing, the 2 phases I'll be discussing in this video. First one is lag phase. Now the growth curve basically looks like this. This is the x-axis, this is a y-axis. Okay, this is time and this is the log growth scale. Means this is basically counts the logarithm of the viable cells present in the batch culture. Viable means live cells, dead cells not. So there are no dead cells right now present in the media, means the liquid culture or the broth. And this growth curve measures the number of viable cells present. Remember this thing, viable cell means live cells. Okay, now come to the lag phase. What is the lag phase? How it looks like if we want to draw in the growth curve. This is a time, and this is the log, the OD values, which we have to measure with the help of spectrophotometer. So the lag phase usually looks like this. It starts like this. Why? Lag phase, basically there is a lag when we introduce any kind of a bacteria, mainly a pure culture of E. coli. Let's say a pure culture of E. coli. If we introduce it into a nutrient broth medium, then first it will need the time to adapt or to understand what are the nutrients present in the nutrient broth, so that it can accommodate accordingly the enzymes or like that. Because a very old culture of any bacteria like E. coli, it's a very old culture. We have to store it in fridge or like that, preserved it at 4 degrees centigrade. Now we need to measure the growth rate of the E. coli. So we first have to add the E. coli into a nutrient broth. Now this old culture of E. coli needs ribosome, huge amount of ATP, different type of cofactors, enzyme, to use the fresh nutrient broth for their growth. Once these type of factors, they are in adequate amount, then the E. coli cell starts to replicate their chromosome, and finally the cell will divide and increase their number. So lag is basically representing the time in which the bacteria can understand what are the nutrients present, what are the environmental conditions are present, in which it has been introduced now, to revive the culture. So this is the lag phase. Hope you like this video. If you like my channel, you can subscribe. Thank you so much.